This is not just your typical medical lab.
These are the epithelial cells that were extracted from a urine. This is actually a healthy control samples, not a specific patient. And once we grow them under the microscope, it takes quite a lot of time, about 3 weeks up to a month, that they are actually fully grown.
What would have been called science fiction is now actually reality.
We are creating brain tissue from urine. That's a few thousand of cells.
Do they move?
They're not. They're actually stuck into the bottom of this plate.
Wow.
It's hard to imagine, but the team at Itay Beyond is creating a brain out of sloughed-off organ cells that usually end up in the toilet.
Those are mainly secreted kidney cells or liver cells that are naturally being secreted out, and we have the ability to take them and reprogram them to become any other type of cell of our body.
What's amazing about it is that they actually have neuron connection between the cells that allow them to be electrical active.
What we're doing is to take them to a living brain tissue that contains neurons and other types of cells. This brain tissue is functioning. It has electrochemical signals like the brain that we have, the original brain that we have.
I take these organoids and I place them on a multi-electrode array, which has 60 electrodes that go out from it. And then I can record the resting state potential of this organoid, which is basically a brain, which is basically a small brain that has the same activity as the big brain.
It may be hard to wrap our brains around it, but the results are mind-blowing.
Measure these electrochemical signals to compare them between brain tissues that we grown from control group and to patients with ASD, autism spectrum disorders, or epilepsy, or schizophrenia, and to see the differences.
We have 60 channels. Each of them is doing electrical recording from the organoid, from the brain tissue. And all these small spikes are actually electrical activity. This is how the organoid speaks.
In this case, the organoid is grown from cells of a patient with epilepsy.
All the electrodes fire together, which resembles what happened in epilepsy.
What do you do with this information?
So now when I have some biomarker of some abnormality, now I can start testing drugs and see which drugs help to restore the activity to the normal level.
And that's the point. All this sci-fi is not in vain. This scientific advancement is meant to be a more cost-effective way to test existing drugs and to potentially help discover new drugs that treat a series of psychiatric and neurological disorders.
Usually it takes about $1 billion, sometimes more, and about 10 years to develop a new drug. Companies don't always know which drug would be effective at the end of the road. They need to wait until the end. We have a prediction method that can help them predict before which drugs are more effective.
I'm very happy I'm not working with mice anymore. Working with mice is not a human, okay? Everything that is FDA approved according to mice, it's not the same as what happens in actual humans.
Mice and rats don't have the same brain structure, brain biology. For example, the most developed area in our brain as humans is the frontal cortex, prefrontal cortex. Mice do not have the same prefrontal cortex. Neurological and psychiatric disorders are usually starting or manifested in the prefrontal cortex.
I developed a Python game, which we can we put the organoid inside the multi-electrode array, and then we give a stimulation on the right side or in the left side. After that, we record the activities, and we get an answer from the organoid. If the answer matched the side that we gave the stimulation, we give him a positive feedback.
What? A video game that's actually played by the organoids themselves?
So the positive feedback are strengthen the organoid synapses, and the organoid actually learns what it's supposed to do. We want to show that the brain organoid from the control group is capable of more learning than the organoids from the other groups.
It just goes to show that by putting on their thinking caps, these brainiacs have big dreams for the near future.
Each drug and compound that we give in early stages, not only for autism, but also for Alzheimer or schizophrenia before the symptoms, we know that it puts the brain and the person on a different trajectory of disorder, which is much milder. In autism, it also has been proven a lot of time that if you can recognize it before the age of 2, and you start even with enrichment, environmental enrichment, behavioral treatment, it can be very meaningful in later stage of life.